Very good. All right. Welcome. In a very Tristan manner, we are fashionably late because we were still, drinking tea, and then we have to go get the laptop. Bad planning. I haven't hosted a sandwich for a while, so yes, bad planning. It's on me. But I'll invite you drinks after the talk for free, whoever wants to come. And hopefully that, balances. It's with great pleasure that we welcome Professor Peter Ulric, who is now visiting us for 1 day, but hopefully he will come for maybe a few hundred days next year, if he managed to get rid of his director's hat of the psychology in Dartmouth College. We wish you best of luck with that. Other than that, excellent scientist, and we're lucky to have him, and we're really happy that he decided to visit. For those who are more cognitive neuroscience hardcore, you will know him from, I don't want to say this, early and mid-level perception and cognition mid-level visual neuroscience, but I'm always a bit, and some other people might know him for things more related. Shall we say, neural causation, right? And I will
this and that.
But that's my interpretation, and without trying to give you a long list of achievements and all of those things, because I never do that. You can Google that bit. I can say that so far it's been a great day chatting with him, and we hope that have a nice hour of talk and then half an hour of discussion, if you're still brave enough. Hopefully you will. And if there's anything that you needs clarification, of course, ask during the talk. But if you have a pressing question, keep it, until the end. Welcome.
Well, thank you, Tristan. Yeah, hi everybody. Thanks so much for it's lovely to be here. I just flew in from Boston last night. So if the ideas are good, you can attribute them to me, but if they're bad, it's jet lag, right? And, so I just was pondering what I should talk about, and I thought, well, I just talk about some things that I find interesting initially. So I will talk about, first I'll talk about some ideas that are in these 3 books. The first one on the left there is really about the neural code. So I'll talk a little bit about the neural code, and then I'll talk a little bit what's in the last book about the nature of imagination. So the talk really has 3 parts. The first one is some ideas about the neural code, the nature of causation among neurons. And then I'll talk about some nice phenomenology experiences. We'll look at some interesting effects. And then I'll talk about some fMRI data concerning the nature of deliberative imagination and volitional control of operations within working memory. But we'll focus first on issues of neural coding. And so I just want to raise this question of, how do neurons drive other neurons, right? So here's some data by Stefan Troya's group, where this is actually an MT neuron that has a fixed receptive field, say here, and there's some moving stimuli, but we won't talk about the stimuli, and the monkey's fixating here. So you can put whatever stimuli you like within that receptive field, and there's different parts of the receptive field. And you can have the monkey attend, in this case, to the left stimulus, and you'll get this pattern of activation. So this is a heat map of spike rate for a given neuron. Now, if you have them attend within the same receptive field to the other one, you get this heat map. And you can take the difference, you get something like this. So something is influencing this single neuron as a function of where the monkey is attending. Well, what's actually allowing this to happen? It cannot be the case that new axons are forming because they're fixed. So it has to be something like changing the responsiveness of the neuron to its inputs. Because in both cases, the stimuli are the same. It's either attending here or attending here. The stimuli are the same, so this change in the responsiveness of the single receptive field to identical bottom-up input is affected by attention. But how does attention work such that this neuron's responses will change? Well, I think it has to be driven by something other than changes in axonal inputs. It has to be something like changes in responses to synaptic inputs and probably changes in synaptic weights. And so something like acetylcholine is being projected down from some brain area, like the basal forebrain, which is influencing this neuron's responsiveness to identical bottom-up inputs. So, a very common metaphor for the brain is it's a bunch of connectivities driven by these white matter tracks. Here's an actual brain. Our group and other groups have done DTI, effectively mapping out similar connectivities. And a metaphor that sort of drives that kind of way of thinking is a highway map. If you want to know, we're connected with what, you look at the axonal connectivities. And that does tell you a lot, right? If I look at some country, say England, and I wanted to know, what's connected with what, I could look at the highway system and say, well, you've got a big highway between, London and Manchester. So there's probably a lot of transfer between those areas, but really there's a lot that's missing from simply analyzing a highway map, right? Any individual car can follow all kinds of different paths. I think more that this metaphor can also blind us. I think metaphors are, both powerful and dangerous, right? Metaphors, I remember I was teaching psychology 1 to a bunch of undergraduates, and I was saying, a neuron's a lot like a toilet, right? There's a threshold, and you pass the threshold, and boom, this, it's a ballistic process, and, and I thought, wow, threshold, and there's a refractory period, and then after class, a student said to me, but Professor, what's the little poop that goes down the pipe? And I said, oh, there's no thing that goes down the pipe, it's a disturbance, right? So this metaphor blinded us, right? I think that the metaphor of the brain is simply a matter of connectivities is useful but blinding. I'd say a better metaphor, it's still imperfect, would be something more like this map, which is the train map. Now, trains are interesting in that the tracks have these interesting shunts, such that a given piece of track might form part of one connectivity or a different connectivity. You might call them epiconnectivities. And depending on the setting of those very tiny shunts, the outcome of information transfer or train transfer in this case would be radically different. So I could look at it, in this case, is the train track map of the United States, and I want to connect, say, New York to San Francisco. Well, I can do it this way, or I can do it this way. And for that one bit of track there, say in Ohio, it's part of two different epiconnectivities, right? So the danger of thinking of connectomics is that you're missing out on epiconnectomics, where a given neuron can be part of many neural circuits, depending on its synaptic weights, which would be the equivalent of these shunts. So in asking ourselves what the neural code is, really, I think we want to focus on how causation works among neurons. How does one neuron drive another neuron? And, or conversely, how is a neuron influenced by presynaptic inputs? Now, for this, I think it's useful to just go to philosophy and look at what work people are doing recently on the nature of causation. So here you have Judea Pearl and James Woodward, who in the 90s and early 2000s have really, I think, done some of the best work in philosophy on the nature of causation. And they have come up with something which might be called a manipulationist notion of causation, in which I think they're effectively describing what scientists already do. You screen off effectively all the irrelevant variables, and you vary some variable of interest, and you see its downstream effects. And they have formalized this. And I would argue that this is not this manipulationist notion of causation is not wrong, it's just incomplete. And it's very much rooted, I would say, actually, in something like a Newtonian conception of causation. So Newton's metaphor might be something like billiard balls. This billiard ball hits this billiard ball, and if you took that metaphor of causation as collision or causation as impact, well, I think a lot of people in neuroscience have an implicit or a quasi-Newtonian conception of causation in the brain, where this action potential drives that action potential. And that's certainly very important, but it's not the entire story, because so in classical causation, Newtonian causation, A drives B. A might have certain necessary qualities or even sufficient qualities to drive B, but given those necessary and sufficient conditions, B has very little say in the matter of what it does. If you hit billiard ball A hits billiard ball B, given these physical qualities like mass and momentum and so forth, billiard ball B has to do this, which gives rise to a sort of bias towards a deterministic view of causation. Now, what I want to bring up is a different notion of causation, which is criterial, in the sense that B now sets certain informational criteria that have to be met by A in order for B to be released. So imagine a very weird billiard ball game where billiard ball B says, okay, well, if it's even and striped, if that's true, I'll do this. But if it's solid and odd, I'll do this. But if it's, solid and, an 8-ball, in that case, I'll do nothing, right? So you can set up essentially informational if-then statements. And this would give rise to a very weird and non-Newtonian billiard ball game. So I want to argue that this kind of causation is very central to what neurons actually do. The standard view of causation in the West in general is quasi-Newtonian, and it's very much rooted in the notion of an implicit metaphor of local impact. So there's a bias in the West towards reductionism and viewing causation as sort of feed forward, whereas in other cultures there's a bias towards other things, like not reductionism but holism, right? Just as an aside, imagine it's 200 BC and some soldier gets hit in the ankle by an arrow and they find that their toothache went away. Now, given the bias, I think Galen or whoever would have said, well, that has to be a coincidence. Whereas in China, that has a holistic version of causation, where things are more like a bubble. You poke it here, it has an effect here. They'd be like, well, that can't be a coincidence. And they might go off and discover acupuncture or something. Anyway, so now the causal efficacy is driven not solely by A as input, but by B's criteria for firing. And those criteria are typically informational, and those criteria can be reset. So an action potential might come along, and it actually might not drive the next neuron to fire, but it might reset the criteria whereby that neuron will fire in the future. And so all of us, all the time, are resetting these conditions for the firing of neurons at many different levels. But it could be very high level, right? I can ask you a question like, think of a woman, poof. Think of a woman politician, poof. Think of a European woman politician, and then you might have said, I don't know, Margaret Thatcher. Well, why didn't you say Victoria May or Angela Merkel? Well, that might have been truly random. And then first of all, then the question is, where does that randomness come from? Is it amplified ultimately from the quantum domain? If so, how might that work in the brain? So I've been very interested in the neural code as this combination of both action potentials, which are certainly necessary, but not sufficient, and rapid synaptic reweighting, and also long-term synaptic reweighting in terms of learning. So I think, there's a sort of yin and yang to these two kinds of causation. So I want to, rather than a highway system or a train track system, I'd like to put forward a couple of other metaphors, and, they're each blinding in their own way, but, I want to argue that the neural code is not just an action potential code, like a Morse code or something. It's also a synaptic weighting and reweighting code, so that, in other words, information is not only transferred via action potentials, it's also transferred via synaptic reweighting. Now you might say, well, what does this mean? Well, here's a metaphor again. So I'd say this is a better metaphor, which would be something like this. So if I were only to measure the energy passing through your vocal system, so I would measure your vocal cords, I would get which alone is not sufficient. I have to also filter that energy passing through the system with a silent filtering imposed by the change of the shape of my mouth. But if I were only to measure the shape of the mouth in the absence of that, I would get this. You need both. You need both the yang and the yin. You need the energy passing through the system, the action potentials, and you need the filtering imposed by these very dynamic synaptic weights, which are functioning at an incredibly fast timescale, at least as fast as the timescale of thought and speech. So the difference between, say, ah and ooh is not because of the energy passing through the system. It's because I shaped my mouth. Right? So there's this interaction between these two types. And you can talk about causation at both levels. Action potentials, but also rapid synaptic reweighting. So I think that the Newtonian conception of causation is really rooted in a sort of emphasis on certain aspects of energy, namely amplitude and frequency, which are no doubt very important, right? So, you can take a given frequency, whatever that is. I can change the frequency, make it slower. And I can also change the amplitude, right? But what nature came up with in the case of biological systems, including brains, is an emphasis not on frequency or amplitude of energy, like not the inertia or momentum or impact power of the input, but on phase. So what is phase? Phase is about the relationships of two or more things. So two things can be in phase, or they can be completely out of phase, or they can be 90 degrees out of phase, or they can have no phase relationship. So there are different kinds of phase relationships. I would say two of the most important are temporal and spatial. So temporal phase, for example, might be events happen simultaneously. That would be one kind of phase relationship. And I would argue that in the case of neurons, simultaneity is of the essence of what makes neurons respond to their inputs. And also shape would be an example of a phase relationship now in space, right? So life kind of, I think, of course, didn't discover phase, but it discovered, I think, causation based upon phase relationships among inputs. And that would be true all the way down to the level of receptors. I mean, back in our bacterial ancestors, these receptors would have evolved to be responding to different forms of inputs. This happens to be an example of an NMDA receptor, it's a glutamatergic receptor that's very important for neurons transitioning from a sort of tonic to a phasic, or from a non-bursty to a bursty type of firing. But this is very sensitive to simultaneity. You need to have three facts that are true. You need to have glutamate binding to the AMPA receptors, which has already depolarized the postsynaptic neuron. You need to have glycine or D-serine binding to the NMDA receptor, which is released by certain kinds of astrocytes, which can effectively, almost like a fuse, turn that off, the opening of that receptor. Glutamate, glycine, and then postsynaptic depolarization. If all those things are true, then the NMDA receptor opens and allows calcium to flow into the dendritic spine, which leads to a transition in the kinds of responses of a neuron. But that would be phase in terms of the shape, what binds to the receptor. So what would be an example? So take another receptor like the serotonin receptor, the 5-HT2A receptor. It's responsive to the shape of molecules, namely serotonin. But it turns out, by chance, I think, you have these other chemicals that exist in nature or are created in the lab, like LSD, DMT, psilocin, mescaline. They just happen to have the same shape at one portion, and that's sufficient to lead to the opening of these serotonin receptors, which has, hallucinatory effects. So there's many ways to meet that assessment of shape, which is one form of phase relationship. I'd argue that neurons are very sensitive to a different form of phase relationship at a much higher level, namely the simultaneity of the arrival of inputs subject to filtering by synaptic weights, right? So if synaptic weights are set to 0 to a dendritic branch, it doesn't matter what inputs you get. If they're open, if dendritic weights are changed, perhaps because of the opening of channels, then that will lead to have an effect on the depolarization of that postsynaptic neuron. And then phase relationships are certainly important at the whole level of whole organisms, right? So if I have, take this lamprey, well, I know that, this neuron fires, and if this neuron then fires and fires and fires, the thing will move like this, which allows it to swim. Now, if I mess up those phase relationships, I'll end up with an epileptic lamprey, and it can't do anything, right? So phase relationships really were emphasized, and an assessment of the inputs in terms of those phase relationships became very central. Yeah, so in this book, I really zoomed in this question of the neural code, and I came to the conclusion that it's kind of Wittgensteinian in the sense that he, who was here actually, as you know, in his later work, focused in on the question of what counts as anything. Like what, in this case, I drew a bunch of, there's a photograph of people from Lapland, right? Well, what makes someone look like a Laplander, right? Well, you might say, well, they all have dark hair. You'd be like, well, actually, that one's blonde. Oh, okay. Well, they all tend to have this kind of nose. Well, that one actually has an aquiline nose. Well, they all tend to be short. Well, that one's tall, right? So his point was that there are no necessary or sufficient conditions to count as a member of a class. That class could be a bird, a game. What seems to be the case is there are these criteria. Let's say there are 10 criteria to count as looking like a Laplander. Well, if someone met all 10 of those, you might say, well, they look like a prototypical Laplander. But let's say they meet 5. Well, that person might meet those 5, and that person might meet those 5, and that person might meet those 5. Well, they all end up having this set of family resemblances because they meet some degree of those criteria. So he didn't care about neurons or even think about neurons, but I think neurons actually are assessing their inputs rather criterially in terms of the informational parameters they happen to be sensitive to. Right. So one thing is, how do neurons do this? How do neurons assess the informational value of their inputs? Oh, here he is. I just had to show this cool picture of Bertrand Russell when he was young, and around this time in 1906 or something, this young Viennese engineering student shows up. And anyway, he's buried somewhere around here, and I would love to go to, if I do a sabbatical here, I'd love to go look at this grave. Anyway, certainly a great mind. Do you recognize this street? I wouldn't know, but that's Wittgenstein there, and that might be G.E. Moore, I'm not sure. All right, anyway. Right, so let's, this is sort of the idealized picture of a neuron that we get in textbooks. But real neurons, especially pyramidal cells, are much more complex than this picture. And we know this because, Golgi discovered the silver stain, which was then exploited by Ramon y Cajal. And he spent the next 40 years looking through his microscope. And he was looking at, say a pyramidal cell like that one up there, and he would see on the apical dendrite shown at the bottom right there, these little sort of hairs on the axons, which are dendritic spines. Now, if you see such little bumps on an axon, you know that the cell's very likely taking glutamate as its input, and there's likely, it likely has NMDA receptors there. It turns out that NMDA receptors, unlike AMPA receptors, allow calcium to enter the cell, and calcium is double positive, so it's highly depolarizing, but too much calcium is very poisonous for cells. So these dendritic spines seem to encapsulate the calcium from getting necessarily too far into the other parts of the cell. Anyway, there's this beautiful work by Lichtman, now at Harvard, that I think what they did is they took a cubic millimeter of cortex and they just sliced it incredibly thinly, and they mapped out that 100,000 neurons and how they're actually related to each other. And his work is very beautiful. And they end up with these seemingly impossibly complex this would be a pyramidal cell. And they're showing all the axons feeding into that pyramidal cell on the left. On the right, they've removed those, and they're showing in the light green there the synapses. So you can see a pyramidal cell is this very strange and seemingly incredibly complex structure. Why do we have such complexity at the level of the pyramidal cells? The pyramidal cells are about 70% of the neurons in the cortex, and they're glutamatergic, they're excitatory, and they're surrounded by all kinds of other cells, glial cells of many different sorts, but especially inhibitory interneurons, for example, basket neurons and so forth, that are essentially modulating the responses of these pyramidal cells. Why would this have evolved? This incredible complexity. This is radically different than what neural nets look like. If you look at the neural nets, CNNs, ANNs, RNNs, they're all built on this model of neurons as points that you can change the weight on them. But actual neurons are radically different than anything like what neural nets are doing. Okay, well, one form of phase relationship, I said, is simultaneity. And it turns out that simultaneity is central to detecting coincidences, the coincidence of the arrival of two inputs. Now, you can imagine, how are you going to create a system that's detecting coincidences? Well, one thing is you have to essentially erase history. Because if you detect something and it stays on too long, this other thing arrives later. Well, they would not be simultaneous. So you have to create a neuron that has essentially no memory. And so what has evolved are these beautiful pumps, sodium ion pumps, that are upon depolarization, you might have, an excitatory, so from your point of view, an excitatory postsynaptic potential, but then it decays because of those pumps. So once you get back down to baseline, it's as if that spike, that excitatory postsynaptic potential never happened. And you could have hyperpolarizing ones, inhibitory postsynaptic potentials, and then decay back to baseline. But let's see, going back to neuroscience 101, you know that neurons have a membrane potential, a difference in, essentially charge across the membrane. The resting potential for most pyramidal cells is on the order of like minus 70 millivolts. And then you stick the electrode in at that vertical arrow there. And if you zoom in, can you see my mouse? Yeah. Here's a bunch of excitatory postsynaptic potential. Boom, boom, boom. And if you reach the threshold for firing, let's say that's minus 55, you get this incredible action potential. And then you have often a little refractory period. So how do you get this kind of summation given the, this descent to baseline because of these pumps? In this case, sodium-ion pumps are getting rid of the positive charges. They have to arrive within a window. Now, it turns out that temporal integration window for a typical pyramidal cell is on the order of about 20, 25, 30 milliseconds. Now, interestingly, 25 milliseconds. Now, why, if you divide 1,000 hertz by 25, what do you get? You get 40. So you might think, well, 40 hertz might play some special role in the simultaneous arrival to pyramidal cells. That's another conversation about the importance of this domain. But this is probably very related to the integration window of pyramidal cells. Right, here's your firing threshold. EPSP, IPSP, action potential. So one way to have your inputs arrive at the same time is synchrony. Have all your inputs from the previous layer arrive at the same time or within this integration window of, say, 25 milliseconds. So synchrony is almost certainly going to be a very important part of neural causation. That's one way to do it. Another way to drive a postsynaptic neuron is not through synchrony, but through one neuron firing like crazy, right? So if one neuron bursts, then it might create, 4 or 5 EPSPs within the integration window. And again, the postsynaptic neuron might fire. And both of these modes of communication are essential, I think, to driving neurons, to driving postsynaptic neurons, to neural causation. So the whole system seems to be set up, we could even say the mantra is something like coincidence is, or coincidence is not a coincidence. It's looking for, among all the noise of its inputs, things that are happening simultaneously. When they arrive simultaneously, certain things might happen. So let's say I have a dendritic branch. Well, the branches, here's the soma, then you have a branch in the basal dendrites, and it forms a Y junction, and then this might form another Y junction. But let's just consider one Y junction. So this is a dendritic branch, this is another dendritic branch, and so forth. Well, these are compartmentalized in terms of dendritic compartments. The system might be set up to depolarize this branch only if this branch and this branch are both simultaneously depolarized, and if not, it won't depolarize. Now, it turns out if you set your system up anatomically like this and have a simultaneity-based code, you can actually carry out all kinds of operations, logical operations, like, right. So now imagine if you can change the synaptic weights such that you can turn the gain up on this one or turn the gain down on this one. You're going to change actually the assessment of the inputs because you've changed the synaptic weights on those dendritic branches. So you can actually set up logical gates. You can set up AND gates, OR gates, XOR gates, all kinds of seemingly logical operations in your dendritic tree. So everyone talks about neural computation all the time as if it were some property of the whole brain or neural circuits. I'd say a majority of the actual computation is happening at the level of pyramidal cell dendrites, which is an understudied area, but I think it's growing. And so you can create all kinds of interesting logical gates among your dendrites. And you can change what those operations are as a function of changing the synaptic weight, so that this neuron at one moment is going to become, it's a cat detector, say in the inferior temporal lobe, but I change a certain rates and now poof, it becomes a dog detector. All right. So if you look at this work, I think this might be Charlie Gross or Edmund Rolls, I'm not sure. This neuron in the monkey equivalent of the fusiform face area responds clearly to something like faces, but it's not just faces, right? I mean, E is a blockhead, it doesn't like that. It doesn't like the eyes alone, it doesn't like the mouth alone. It seems to have particular conditions that need to be met. One is a roundish background, and then something like two eyes and a mouth simultaneously. And if those conditions are all met, if those truth conditions are met, the neuron will fire. So you can think about, I can set up a neuron to detect faces, depending on its inputs. And this does seem to be part of how things like faces are encoded, right? You have no face detectors in the retina or V1, but you can wire them up effectively genetically or through experience. And then, it can lead to these really kind of weird and cool experiments. This is stuff done by Quiroga, Itzhak Fried, Christoph Koch back in the early 2000s. In this case, they had epilepsy patients, and they were sticking electrodes into the hippocampuses of those patients. And they did things like show them a movie, an episode of this TV show called Friends that has this actress, Jennifer Aniston, and they would then measure the response of those neurons to stimuli. Those neurons seem to respond to what they had experienced. They had just watched an episode of Friends for the previous half hour. This neuron now seems to respond to Jennifer Aniston and not Julia Roberts and not other things. And people thought, oh, well, that's weird. Why should we have neurons tuned to Jennifer Aniston? Well, they're very dynamic and they can change. So they then did other experiments where I think in one example, Itzhak Fried at UCLA showed patients, ECOG patients, an episode of The Simpsons. And in that episode, different things happened. And every time Bart, this terrible boy, he would, the neuron would seem to become a Bart detector, right? And then they had the clever idea of having people recall what had happened. They said, okay, close your eyes and tell us what happened. And then they'd say, oh yeah, Marge was the mother. Marge was in the kitchen, and then Bart came in the door on his skateboard, right? And so it's remarkable, right? So this neuron, which previously, maybe an hour before, had nothing to do with Bart, was now responding to Bart in the hippocampus. So these are very weird results, and some of the results have gone even further and are even, I think, more important and even weirder. They have come up with the idea of concept neurons. Well, they'll find some of these neurons in the hippocampus that respond to something like Oprah, but they'll show a picture of Oprah, brr. They'll show the name written, Oprah, brr. They'll play her voice, brr. So it seems to be responding to Oprah-ness per se, and not any particular mode of input, which is a remarkable result. And then later, maybe 10 minutes later, that same neuron is responding to something entirely different. Right? So it's not a hard set tuning, it seems very dynamic. So then the question is, how do you wire up this kind of sensitivity among neurons? So I think that our whole field has to move in the direction of the neural code as not just an action potential code, and we're hoping to find the Enigma or Morse code of firing rates or patterns, but really to zoom in on dendritic weights and how they're dynamically changing on a millisecond time scale. And this is a really hard problem because we don't really have the technology to zoom in on and measure these synaptic weights. And yet, if this is half of the story of causation among neurons, ultimately I think we have to go to this level. But I want to just argue that this kind of tuning property of neurons, namely that they respond, we might even say equivalently, to many different forms of input that meet their criteria, say of Oprah-ness. Right? This applies all over biology, but especially to our subjective lives, right? So take the case of, me, right? So when I was looking for a wife long ago, I had some set criteria, which were presumably, being heterosexual, I was looking for a woman. Now, does that mean that there was one unique woman in all of human history that would have been for me? I mean, I told her that. But in fact, probably not. Like, had I turned left that day rather than right, I would not have even met her, right? And I, certainly was not going to marry any of those things. But, then the question is, what then led me to choose her versus all the other women I could have married? Certainly had criteria, I would say, had to be funny, intelligent, kind, good looking, healthy. I mean, all kinds of normal. I don't think I'm unusual in those, and all of us have these criteria. And, probably there's some criterial satisfaction subject to a threshold, and that threshold can be moved up or down, right? Like, let's say I look for 10 years and I can't find anybody. Well, maybe I'll, and then, and then I meet her and I'm like, honey, you're the one. Anyway, but I think that this kind of contingency is all over our reality, our subjective life, and that, things can truly turn out otherwise under a criterial encoding. Because there's an infinite equivalence class that could have met any finite set of criteria. Okay, so let me just summarize this first, what I hope is a third. Pyramidal cells, it's a coincidence or coincidence detection system at every level, from the receptor level up to the dendrite level and across dendrites and ultimately at the neural circuit level. Simultaneity is really the essence of what the system is sensitive to. It's almost like neurons are listening for their melody among all of the cacophony of inputs. And then when it hears it, it's like, yes, I detected a cat, or I detected whatever. And so this encoding at the dendritic level in terms of simultaneity of inputs, of the inputs that would decay back to baseline, realizes logical operations. And the criterial assessment of inputs, and this can be changed very dynamically, such that neurons can change the criteria of another neuron, or themselves, such that they'll fire to different things in the future. Right? So they not only make each other fire in the quasi-Newtonian notion of action potential A forces action potential B, they might come along and this neuron might have not fired, but now it's tuned to something else than it was prior. Okay, so I just want to touch on something that I find to be some of the most exciting work in neuroscience right now. And this is the work of Matt Larkham, who is at Humboldt in Berlin. He's an Australian. I've never met him, so I'm not, I'm not selling anything. But you zoom in on a bit of cortex, like that little rectangle there, and it's actually something like, people call them columns, mini columns, but it seems like something like this might actually be the fundamental unit or brick from which we build the architecture of our brains. That it's not the neuron per se, it's this neural circuit that you see again and again and again all over cortex, almost like a honeycomb, like a mosaic. Now, a column or mini column, in V1 might be ultimately tuned to something very simple, like a orientation. Whereas, a mini column, if you take Tanaka's work at RECON seriously, it could be something really complex, and maybe it might end up being further down the temporal stream, something like a Tiger detector at this neural circuit level. So this is like 80 to 100 neurons full of pyramidal cells and inhibitory interneurons and other kinds of cells. If this is the unit, the kind of the fundamental functional unit of the cortex, another thing for our field to do is really analyze this level of functionality, not just the neuron. So what Matt Larkham is doing is he's arguing that simultaneity is fundamental to consciousness. And so, pyramidal cells have this very unusual architecture where in typically layer 5 or 6, but let's say layer 5, you have all these basal dendrites, but then you have these apical dendrites that reach all the way up to layer 1. You think, what a weird organization, If I had input all the way up here, well, if I were to rely on passive diffusion of potential, it wouldn't even be able to depolarize, certainly at the level of the axon hillock. So why would I have this crazy long apical dendrite? So his argument is that first, there's not just passive diffusion of potential down the apical dendrites. There's actually active propagation. And his argument is that there's top-down input here, there's bottom-up input here, and when the top-down and bottom-up input are simultaneously arriving, that cell will do something, or it won't do something. But the simultaneity is key. And where they have the red circle there, it turns out that that is where you have certain higher order, he calls it HO, higher order thalamic input. You can shut down the communication between the apical and dendrites and the soma, and that typically leads to unconsciousness. So a lot of the anesthetics operate by knocking out receptors there. So what's labeled as C here is if you knock that out, and there's lots of these different anesthetics, very diverse in their chemical nature, but they seem to have in common knocking out that intermediate branch such that the apical dendrite can no longer communicate with the soma, and that leads to unconsciousness. That's his argument, not mine. So, but that is another higher level form of simultaneity, not the dendrites, you know, if this and this, then this, but now we're talking at a whole cell level of simultaneity, and then we can talk about other forms of circuit level simultaneity. Right? So this is his argument for how anesthetics work. And you know, people going back to the 90s were discovering actually, you know, the apical dendrites seem to be propagating something like action potentials, but they're not going from the axon hillock out to, you know, other parts of the brain. They're propagating from the soma up and then sometimes seemingly back. And so one argument is that these apical dendrites are playing a role of simultaneity detectors in the sense that you send something up, if that's true, and that arrives at the same time as this top-down input, send the signal back and keep your, say, NMDA receptors open for longer down at the basal dendrite level. Anyway, I just wanted to raise that. I think the general problems for our field are enormous. If we're serious about thinking about the neural code, not simply in terms of the yang of the energy or action potentials flowing through the systems, but in terms of the ways in which simultaneity is used as a way to impose informational criteria for firing, we need to really look at the dendritic level, and we don't really have the techniques for that yet, but something for future generations to do. Yeah, anyway, I'll just skip that. Okay, so now I just want to totally shift gears from the neural code, which is something that we should all think about, but it's not solved. And we haven't, I would say, yet had our Darwin to present an overarching set of ideas that unify all of biology's, in this case, neuroscience, or even our Watson and Crick. You know, we haven't yet. So there's one of you in this room might be the future Watson and Crick, and I hope so. And if you figure it out, email me, and I'll take you out for coffee. Okay, so I just want to show some cool effects. So a lot of what we do in the lab is looking at various forms of conscious volitional control of subsequent bottom-up inputs. So let me just show you a couple of examples of this. Some of these are very well known. Let's take this case. Okay, so all women in this room, close your eyes. If you're a woman, close your eyes. If you're a man, keep your eyes open. Okay, now do the opposite. All men, close your eyes tightly, and all women, open your eyes. Okay, now everybody, open your eyes. Now, if you, Ben, what do you see? Say it. Donkey. And women, what do you see? Seal. Okay, so what did I do? I showed one sex that and one sex that. Donkey. So then by priming you, you know, with this ambiguous thing, one sex saw a donkey and one sex saw a seal. Now, that's a good example of, you know, semantic priming. How did that happen? Well, you weren't growing new axons, right? This had to be, I think, priming the system in terms of probably synaptic weights. That's just one example. Here's another example of a kind of top-down effect that we're looking at now, where if you fixate any of those dots, let's say fixate the one in the middle, if you volitionally attend to any of the disks, you'll find that it seems to become subjectively darker, more opaque, and it shifts to the back. So look at, say, fixate the central dot, and just volitionally choose to attend to any of those disks, and you'll find that you have some volitional control over the subjective experience of its opacity, darkness, and depth. So how does that work? How does volitional shifting of attention influence, you know, the outcome of processes that lead to your conscious experience? This would be another example where whichever disk you attend, not just fixate, you're fixating the central dot, should, in this case, become more opaque, lighter, and go to the back. So we're very interested in the lab in these kinds of cases where there's seemingly volitional control of subsequent conscious experience. This would be a color case where, if you fixate the central dot in the gray zone and you attend to, say, the blue disk, it looks rather like there's a blue disk behind these transparent filters. And if you attend to the, say, green, it looks like there's a green disk of uniform color behind these transparent filters, even though in fact in the gray zone in the middle, it's always gray at the bottom, at the level of the input. Okay, so another example of top-down control that we've been interested in is this very basic example of apparent motion. So given, your visual systems, you probably see this as moving. Who sees it jumping up and down vertically? Okay, who sees it jumping back and forth horizontally? Okay, it's about half-half. Now, I've learned through experience that I have to make it slightly more vertical, the jump, because there seems to be a bias to match within hemifield. In order to have it be roughly 50-50, as it appears to be in this room, I need to make it slightly more elongated vertically to make it, if I made it square, it would tend to match overwhelmingly vertically. Now, if you're seeing it, let's say, jumping vertically at this present moment, you can cover the bottom two with your hand. And then that would force it to match horizontally. And if you see it going horizontally, you could also cover the left two. And so you're able to bias it now to match the other way. Now, some of you might have strong attentional muscles, and you're able to volitionally choose to see go clockwise or counterclockwise. Or you can volitionally choose to see vertical or horizontal. Can anybody do that? Okay, good. So I wanted to start the attentional Olympics, but I didn't know how to score it because people might just lie. But thinking about this is actually something we all experience. It's when you look at the lights on top of a police car that has stopped you, and as you're getting out your wallet to get fined, you're thinking, how strange! I see the lights jumping back and forth, when in fact I know cognitively that there cannot be any light bulbs jumping back and forth. Why do I see motion at all? Well, it's pretty strange apparent motion, right? So something appears, and then later something else appears here. Well, the system seems to have the bias to say it's kind of one thing that shifted here, but it cannot be the case that the inference of motion started when this first thing appeared, because a second one might never have appeared. So it has to be the case that within a certain buffer, this appearance of the second one gets inferred to have been that one, and you infer a perception of motion. This seems to imply that there's some kind of buffer prior to the conscious experience of seeing the motion, during which events are compared over large spatial distances and temporal distances. And so what is the duration of that? Well, we did a kind of experiment, a very simple experiment, in which we created these apparent motion quartets, zoomed in to a tiny little, so they're very tiny, less than 1 degree, right around fixation, and you just have two dots that go expanded. Boop, boop. We'll call that time 0. And people have to subjectively report whether they saw vertical or horizontal. And we might just look at whether people can do that, and we can impose a kind of catch trial in which we force it to match, say, vertically, boom, boom, boom. Or we can force it to match horizontally, boom, boom, boom. So those are catch trials. And among subjects who are completely honest, let's say 100% honest, that they saw vertical in this case and horizontal in this case, we might look at their data. In the case where there was no intervening test value, just boom, boom. Now that's a square. It was in fact a square. And so we can then say, they report I saw vertical or I saw horizontal. Now we can ask them the question, can you bias that volitionally, right? So we might do an experiment like boop means try to see it go vertically, or boop, or boop means try to see it go horizontally, and people can do this. Now it's not surprising really that people can have some subjective control over whether seeing it jump vertically or horizontally if you give that command with a tone before time 0, which is the switch. But what's interesting, okay, so first we started off, we didn't tell them the meaning of the tone. So they hear the tone, but they haven't been given a command that whoop should mean vertical, or whoop should mean horizontal. So, right, they're basically they're at chance, 50-50 in reporting vertical versus horizontal. But now for the same kinds of inputs, we give the command, if it's boop, try to see it go vertically. try to see it go horizontally, and that command can happen before the switch. Now that's so people can have some effect, but what we notice is that in these data, let's see, after we told them the meaning of the tones, that even if we presented the command after the time zero switch, they had some ability to influence what they perceived. Now that's interesting, right? Because the command, the volitional command to try to see it go vertically happened after the event. And so, in this basic psychophysics, I think in this first case, it was out to 200 milliseconds. And so, and this was a lot of subjects, it was like 80 subjects, and I can't remember what journal this was, it doesn't matter, some psychophysics journal, and the reviewer said that's the backward causation in time, this can't be correct. But we argued, well, listen, no, there's this preconscious buffer between retinal activation and your conscious experience of motion. There's, turns out, 200 to 300 milliseconds, let's say 300 milliseconds of evaluation over, in light of multiple constraints. And an intention can actually zoom into that preconscious buffer and influence the outcome of its operations such that you will subsequently perhaps see what you intend to see. So they had us redo the whole thing with a totally different set of 80 subjects, which we did. And this time we said, okay, we're going to go out even further, out to whatever it was, 350 milliseconds or 330 milliseconds. And again, the same sort of results, that even beyond 300 milliseconds, the command comes in and people are able to influence what they perceive so that there's this preconscious buffer. So we're all sort of phase shifted out, like you see my mouth moving and your conscious experience is not simultaneously with the light arriving in your retina. There's some processing, which might be a quarter to a third of a second, in which volitional operations, like an intention to see vertical versus horizontal, can affect outcomes. Okay, this is just an example of top-down processing. So the story, though, looks something like this. Time zero is retinal activation, the switch from these two dots to these two dots instantaneously. But it seems that there can be some top-down modulation of these unconscious, preconscious operations that then affects subsequent conscious experience. Right, and so now I'm going to jump to some MRI. It's a kind of different question, but in my mind, these things are very related. That having a neural code that's very criterial like this and subject to loose constraints has an effect on how we imagine and how we evolved to imagine. So what I want to argue here is that human imagination is pretty interesting in that we have evolved to have internalized quasi-motoric operations that allow us to do things in our working memory. Okay, so I'm going to talk really about 3 experiments. So first I'm just going to motivate those experiments. This is some MRI. This is coming out next month in Psych Review, which I'm happy to send it to anybody. But I'm arguing that there was a kind of breakdown of modularity in the human mind that led to a particularly human form of imagination that's very different from, say, that of a tiger, which is a much more modular mind, and we can talk about that. So let me just give a kind of a super quick review of human evolution, right? So if you look at human evolution, where the y-axis would be down to 2 million years, well, there's been lots of different hominins, and we are sort of the fusion of several subspecies, you might call them, the Denisovans, the Neanderthals, and so forth. Going way back, 6 to 4 million years ago, there seems to have been a real big climate shift in East Africa, especially where the jungles gave way to savanna, which forced our ancestors out of trees, which really was a tremendous selective force that led first to bipedality. So long before our brains changed in volume, we were bipedal for at least, I would say, 2, even 3 million years. Now, here are some of these. What they do is they give the bones to these forensic specialists who are able to reconstruct murder victims, by putting all the little muscles on and putting on skin. They let these people, these artists, really reconstruct what our ancestors might have looked like. And, this is the rest guys. Of course, they can't know what the soft tissue is like. or the hair is like. So these are best guesses. They can't really know what the nose was like. But if you look at the Australopithecines on the lower and central bottom, their brains were not very much bigger than chimpanzee brains, like on the order of 350 to 450 cubic centimeters. Whereas when you get up into Homo habilis and Homo erectus, you have much bigger volumes, and then in us, even bigger. But things were going on already among the Australopithecines. So here is whatever 600 to 800,000 years ago, you had Homo heidelbergensis, which seems to have given rise to different hominids, including us, but also the Neanderthals and the Denisovans. Have you heard about the Denisovans? I mean, this is so interesting. So about 2000, this is a little aside, you have to bear with me. So in 2010 or so, there was this amateur Russian paleoanthropologist who was going through the caves of the Altai Mountains, and there was a cave that had been occupied by some monk Denis, in the Middle Ages. And he found a little pinky bone, and he said, that's funny, it doesn't look like a Neanderthal bone. I don't know what it is. So he sent it to the Moscow Institute of Sciences, and they said, we don't know what it is. So they sent it to Svante Pabo at the Max Planck for Human Evolution in Leipzig. And this is why some scientists are like, there's great, and then there's like super great. So what he did is he was able to bore into this pinky bone, and he got the DNA, and he was able to decode the whole genome. And then he went and looked at all extant human genomes, and he said, and in addition, Australians are 6 to 8% of that, right? So these people are 6 to 8% Denisovan, which I think is an astounding finding. Anyway, but we're, except for sub-Saharan Africans, we are all, to some degree, Neanderthal. And East Asians and Southeast Asians and Papua New Guinea Highlanders and Australians also part of Denisovan, which is all I think is all super interesting, but that's an aside. Okay, so here we have Lucy at some Australopithecine ancestor back in what is now Kenya, left their footprints. So the first stone tools are found in Kenya, and they were probably made by Australopithecines. And you might say, well, that looks just like a rock, right? But what they have found is that those rocks actually had to be tools because they show evidence of having been napped. And then they had to rotate and nap again and rotate and nap again. So there's some intentionality behind it. And so even though they look kind of crappy, like I can do better than that. Still, the journey of 1,000 miles starts with the first step. And I want to argue that this process of stone tool making, put tremendous selective pressure upon our species or our ancestors, such that those animals, those beings that were better at doing that, could hunt better, eat better, and so forth. So there's a tremendous selective pressure upon that. And in fact, you need to be able to do this in your mind before you hit the rock. You want to do it correctly because you don't get a second chance. If you break the rock, if you can model it in your head and then strike, you can actually have a vision in your head first. I want to make that. And there could be, once that existed, that behavior could be then subject to tremendous selective pressure. And I believe it was. And that ability to model things internally is probably, I think, my guess is that how we got off the ground and having a kind of imagination that is so different from that of a chimp. We were able to form internally estimates of what is likely to happen and then play them out deliberatively and then before doing it. So this is about 2.2 million years ago. The Alduin tool set is still pretty poor, but it's much better than the Lomekwi tool set. Then you have, I think, a real breakthrough with Homo erectus, which is, say 1.8 to, well, they might have been around as recently as 200,000 years ago on Java. They invented this Acheulean hand axe, which was truly beautiful. And here they are. This is the known domain of Homo erectus. What's puzzling, though, about them is that their Acheulean hand axe has hardly changed over space and time for something like 1.2, 1.4 million years. It's really striking, the lack of innovation. So what was going on? Is it something like a bowerbird that has a model in his head of what, or, there's no way, we don't know. Neural circuits only fossils, thoughts only fossils, we can't really guess, but it's striking how uninnovative their tools were, even though they were very beautiful. There's one. Right, so there's a Homo erectus brain. I mean, our brains are clearly bigger. Some people argued that Homo erectus might have made this first art. This is only 5 minutes. Okay, well then I probably can't get to too much of the MRI data. The Mousterian tool set was much better, but still very innovative over tens and tens of thousands of years. And then our species comes along. Oh, this is something that, these are a bunch of stalagmites that must have been made by Neanderthals because it's so long ago. So they were us. We were mating with them, right? But they were seemingly much less innovative than our species. They seem to have even had string. Right, so then our species, the first fossil evidence is in Morocco at a place called Jebel Irhoud. And this is another reconstruction of that person. I mean, this, we don't know the skin color or the hair, so this could be just racist. But their tool sets were a bit not so different from the Mousterian. But later, our species starts doing other sorts of things. I just want to say an aside, if you go back 300,000 years ago, there's all these different hominins alive at the same time, so it was rather like, Lord of the Rings. Our species starts doing really, really different things, like art, what we would recognize as true art. And our species presumably shows up in Central Europe around 45 to 40,000 years ago, and all around the caves in southern Germany, possibly because they came up the Danube and couldn't go any further. You find these remarkable findings like this so-called Löwenmensch, which might be as old as 40,000 years ago, in which was a lion's head on a human body, which doesn't exist in the world. And you see the first evidence maybe of something like symbolic processing. We don't know what this means. This is 80,000-ish years ago in South Africa. Humans seem to have been adorning themselves symbolically with, for example, necklaces. And, we start creating things that are not recognizable to us as art. Some people claim that this little pebble from Israel 280,000 years ago is art, but clearly there's a change in not just degree, but kind, I would say. And then you have this Pleistocene, I don't know what this is, art, porno, whatever, but you have this musical instruments. So something is really changing in our ancestors. And so, can I just do one MRI experiment? Okay, because I wanted to talk about many. What I want to argue is that something really happened in our brains that gave rise to this ability to, that led to our ability to fashion things in our minds before executing them in the world. And central to this was this evolution of the frontal lobe. So even though Neanderthals had brains that were substantially larger than ours, maybe 16 to even 1,800 cubic centimeters versus 1,350 to 1,500 cubic centimeters, our behavior exhibited much more, I would argue, creativity. Changes in brain size, right? So it's really the frontal lobes that seem to, which other animals have, but seems to have really changed in us and ballooned in us. And so something like working memory buffers and the ability to sequence events, especially say area 10, I think led to a very different form of imagination in us than say a chimpanzee. So the argument I make in that theory paper is that a kind of modularity that we have, that our bodies have, became more permeable or broke down, so that a mental modularity that is very prevalent, say, in a chimpanzee brain and mind, where you might have a specialized, fertility assessment detector and a specialized, broke down to the extent that operators that evolved for one purpose could now be applied to operands for which they had not evolved. And so, we already knew that there were, capacities even in modular mind that seemed not to be. So Fodor in his book Beyond Mental Modularity talks about modules being encapsulated, dedicated, parallel, rapid. But even in his book, he says there's other processes that are not parallel, that are more general, not fast, but not parallel, but serial. And attention would be one of those. Attend to this or attend to this across modules. So what I argue is that attention changed such that an object file, which can be thought of as a working memory representation that can hold lots of things like, oh, it's a bird, it's a plane, it's Superman. Well, what changed over that time, the label changed, the location changed, the features changed, but what was held in common is that I was attentionally tracking it. But we don't just attentively track features, we carry out mental operations. And so I've become very interested in the nature of volitional mental operations. And I want to argue that certain classes of mental operations that we are able to carry out gave rise to all kinds of interesting things, like symbolic thought and metaphorical thought and art. But in our field, people typically think of sort of the nouns of the mind as representations. What am I holding in my working memory? But in fact, we're not just doing that. We're also carrying out operations, putting things together, taking them apart, and so forth. And these are sort of, I want to argue, quasi-motoric in the sense that they can involve actual manipulation of objects in our internal virtual reality. And later experiments we did actually make the argument that, when I physically manipulate, I'm using actually some of the same motoric operators that I would for manipulating this as doing it internally. And I won't have time to go over it, but we did MRI studies where we have people physically rotate things in a 4-way classifier, and we have them mentally rotate things, and we can use the physical motoric classifier to decode imagined and vice versa in certain motoric and premotoric areas and other areas. Okay, so I don't know if I don't have any time left. Okay, okay, all right. Well, let me. I guess we should open up for questions. And I had all this MRI stuff, but I clearly talked too long. But I want to argue that when our system changed, affording these sort of the application of mental operations over operands from other modules in a breakdown of mental modularity, this gave rise to very strange things. For example, you could take a fertility operator that was looking for the symmetry and ruddiness of a face or buttocks or whatever, but now apply it to things for which it had not evolved, like flowers and sunsets. You could, there's all kinds of, once you open up these operators broadly, Gestalt visual pattern operators could now apply not just to vision, but to the contents of your memory, and you see patterns across your life. This led both to incredible creativity, because you could volitionally control or non-volitionally affect outcomes in very unexpected ways, but it also made us kind of crazy, right? We're kind of like insane chimpanzees, right? So no chimpanzee will go and say, well, it hasn't rained in 6 months, therefore the gods are invisible gods are angry with it, therefore throw a baby in the volcano or whatever. That's our species, and so our species has this incredible power that the chimpanzee mind simply doesn't, which is the ability to affect. It's almost like we have the somatic and perceptual consciousness of a chimp. In addition, we have this sort of invisible layer of mental models that explain all that. But in this invisible domain of mental models, this bifurcated consciousness, we can therefore do things like infer facts about the world that end up being true. So we have a lot of hits, but we also have a lot of false alarms. So, for example, if I have a lion and I show the lion a lot of zebra hoof prints, I can tell you what it will do. It'll probably do nothing because it doesn't look like a zebra or smell like a zebra. But our ancestors began to form models and say, well, they were probably going this way 2 days ago, and one was injured, and there's probably some savanna over there with grass, and we made a whole story or narrative, which allowed us then to function better than lions, even though we're weaker than lions. All right, well, there's no time to talk about the MRI, so I'll just, I guess I should stop.
We are past the time, but it will be shorter. The more you talk, the shorter the discussion.
What do you guys think? Should I show you quick MRI stuff, or should I? Let me just run through it super quickly. So this is a paper that. So this is. Let's run through the. We wanted to know how this was possible. Like, how did our ancestors create this? How did you download a lion's head and a human body, and then put them together, and then later go make it, right? And so we came up with this basic task, where you had some shape, or you had components, and you had to put them together, or remember them. Or you had to disassemble them, take them apart, or remember them. So there's 4 different things you can do. You can, and so this is what an experiment would look like. People either had to construct a shape from parts, or deconstruct shape into its parts, or simply maintain them. And so looking at certain ROIs, we can decode, manipulation taking things apart versus remember very easily. But we then tried to squeeze more juice out of the MRI lemon by looking at actually mistakes that the classifier makes under the assumption that there's information in mistakes. So we looked at what's called the confusion matrix. So here the assumption would be that the classifier would tend to mistake the putting things together or taking them apart because they're both mental operations, like, versus maintaining, that these should be more confusable. So if you take a confusion matrix like this and you correlate it with the actual patterns of confusion in these different brain areas, you can actually say, oh, okay, well, where are the inputs most similar? Well, the input is very similar at this stage, early on, at the input, whether I take them apart or whether I remember them. And for the operation, I could have, what should be confusable should be manipulated to the two manipulation cases. And then the output, well, this ends up like this. Right. And, I take them apart, I get this, I maintain this, I get this as well. So you get a different pattern of confusion. So we simply ask the question, okay, what parts of the brain allow us to see a pattern, because this is a very difficult task. It takes many, many seconds, where we find this pattern, and we were able to find a correlation between the confusion matrix in certain brain areas. It turns out posterior parietal, DLPFC, precuneus, there seems to be some kind of working memory circuit that allows us to carry out mental operations, and we can actually see it unfold in time. And then I guess I'll end here, but basically we're looking at classifiability over patterns of functional connectivity. So functional connectivity is just looking at two different brain areas that tend to correlate, and we could actually classify those patterns as well. So I think what we're getting at here is, the neural basis of these kinds of interesting mental operations where we put things together, take them apart, and so forth. And I think that's really a central part of working memory. It's not simply maintaining representations or the nouns of the mind. It's really about the verbs of the mind that carry out these operations. Anyway, okay, I'll stop there. My children tell me I'm too chatty, so I'll shut up, and I guess we'll start taking questions. All right.
As usual, those who need to leave quickly can, leave out. If you're brave enough to stay, you can have a nice discussion, and of course, there will be free drinks afterwards if you want to come. So let's open, the discussion. Priority for younger-looking people. Questions.
Stop
I
talking
will
about me.
decide who's younger-looking. Ahead, go ahead. Don't be shy. Yeah, a younger-looking fellow.
Gladly, my hearing's not the best.
In the beginning, when you talked about metaphor,
Yes.
Is it your opinion that the language of code is a metaphor as well?
The language of code. You mean computer code?
Yeah, for example.
If it's a metaphor. Well, I mean, I don't know about code per se, but I think that the metaphor that the brain functions like a computer is a metaphor, and it's very misleading, right? So prior to, if you look at the history of psychology, it starts off with Wilhelm Wundt in like 1879, and he has this very weird notion of psychology, namely it's introspectionistic and kind of reductionistic. So he's looking for the atoms of the mind. What blew people's minds in 1860 was Mendeleev's periodic table. And so Wilhelm Wundt is basically saying, metaphorically, what are the atoms of the mind? And so he would say, graduate students, sit down, here's a red apple, what are the components of what you see? And the student would say, redness and roundness, right? There were two rejections of this kind of psychology, which was rooted in the metaphor of Mendeleev's atomism. One was behaviorism in the United States around 1910, 11, 12, in which they said that's not science. And they eliminated the introspectionism and they kept the reductionism. and they came to view the brain as a input, black box, output. Almost simultaneously in Germany, you had a parallel rejection of Wundtian structuralism, which rejected the reductionism but kept the introspectionism, and that was Gestalt psychology. Now, Gestalt psychology kept going on until the 1930s, but Max Wertheimer and Kafka were Jewish, and so the Nazis almost destroyed Gestalt psychology, but many of them went to the US. Now, the US at that time in the 1940s, 30s, 40s was dominated by behaviorists. So these Gestalt psychologists got jobs in minor universities, not MIT and so forth. So what brought down behaviorism was not actually Gestalt psychology. This kept going until about 1956, the so-called cognitive revolution. And what happened was people like Chomsky, Jerome Brunner, Miller, and so forth, they said, actually there's something in the black box. It's a computer. So the metaphor, sort of a Turing or a von Neumann architecture, so actually the brain's really like a computer. But it's still a metaphor. And that metaphor blinds us now because we still, I think implicitly, are driven by the metaphor of the brain and mind as a computer. It's just not, right? There's no hardware-software distinction in brains. There's no consciousness or no nothing like hunger. There's nothing like software. If there were, I would download, Arabic and speak fluently tomorrow, right? So the metaphor of the brain as a computer, I think, is useful, but also very fundamentally misleading. Basically, it seems like everything that comes along, the next great thing, people say, oh, the brain's just like that. So for Leibniz, it was a mill, and for Freud, it was a hydraulic system, and for Chomsky, it was a computational, like von Neumann architecture. And now somebody's going to come along and say it's just like the internet. Yeah. So anyway, in general, I think metaphors guide us, but often blind us.
Just to clarify, I don't think that code is, in a literal way, instantiated in the brain. Right.
So there's no, I don't. Like a code is something that is separate from the physical instantiation and can be implemented. But no, the brain just has a physical instantiation. So it's not at all like code that can be transferred easily across platforms. And I think that there are aspects of our system that are really rooted deeply in our physicality. So for example, I could ask a question like, why is the synapse on the order of 20 to 40 nanometers? Well, given the temperature of our brains and the viscosity of the fluid between neurons, it turns out, I think Sejnowski and others have done modeling, right? If it were 1 nanometer wide, the vesicle release of the neurotransmitter would arrive very quickly with very low variability. If it's 300 nanometers wide, it would take a very long time, right? So subject to the need to be as quick as possible, but also subject to the need to be highly variable, it turns out that the optimal distance is on the order of 25 to 40 nanometers. And is that a coincidence? Well, I would say, I don't know. But the synaptic cleft, I think, probably evolved subject to the need to introduce variability to neurons such that there's variability in the timing of spikes. And if the system's set up to detect simultaneity at every level, then variability in timing of events will play some role in what counts as a solution to any set of parameters that are set. So there can be the essentially amplification of noise from Brownian motion in the synapse up to the level of spike timing differences, which might play a role in finding more than one possible solution to a given set of constraints.
Good.
Any other questions?
I'm keeping my questions for the dinner, but apparently everybody's coming to the dinner. We should call the restaurant.
Very
young looking.
Yes,
Really young looking.
Yeah, anyone younger than me is young.
Can I try and pin you down to something a bit concrete? So at the start, you said that you gave this example of this MT neuron that responded, that changed its responsiveness to different visual inputs depending on attention, and you suggested that there was prefrontal cortex was sending some top-down signal that changed that achieved this by changing synaptic weights. Is that correct? So in order to do that, you'd have to be able to change. You'd have to be very specific changes, right? You'd have to say, in order to make this single neuron change what it responds to, you'd have to be able to target specific synapses.
Or you could target, the layer below it or the layer below it, So I don't necessarily have to target this particular neuron. I could target its input neurons, which have, say, I don't know how low in the hierarchy, but they have actually physically different positions. And if my attentional map that allows me to spritz acetylcholine into the synapses of neurons that have this location on the retinotopic map versus here is precise enough, this would affect the inputs to this MT neuron.
Right, it's not the MT neuron. It's the you're basically inhibiting or facilitating neurons that provide inputs to.
That's right. But still, I mean, this would be a top-down modulation of bottom-up inputs. Maybe not necessarily at particular branches of that particular MT neuron, but you have to affect. I mean, it's hard to certainly we're not explaining this in terms of axons, right? It has to be something like either changing the weights of the dendritic responses of this neuron or of prior neurons. They're firing properties such that they then feed into this MT neuron differently as a function of where attention is. So
that's not necessarily synaptic plasticity, right? That could just be inhibiting or facilitating neurons that are providing the input.
Right. So there's multiple mechanisms, I imagine, that lead to changes in output of any given neuron.
Just out of curiosity, it seems like the spatial attention and volition, typically people talk about the prefrontal cortex, and you also spoke about it, and it's the region that evolved the most in humans, etc. And my background is in research on memory, where the hippocampus also has its very unique combo of cells that look really different to anywhere else. And I was wondering, if that's part of your thinking of what changed, or is this what do you make of all of those funky cells and their weird operations?
Well, I mean, so
Carefully dismissing the hippocampus now.
I'm not dismissing the hippocampus, but I mean, you can, like in patient HM, you can remove